Well, in order to make RNA, we're still going to use the same ATGC binding principle, but we're going to switch it to AUGC in this case because when making RNA utilizes that U instead of the T. But again, it's the very same principle. So if we look at this image of we start with a double-stranded DNA. Within that DNA, we have the individual genes. The genes are individual instructions for some characteristic or some protein that'll be made. And especially in eukaryotic cells, if you think about the fact that the DNA is inside the nucleus and the ribosomes where the proteins are made are outside the nucleus, there needs to be an easy way to get the information that's in that gene out to the ribosomes. And the way the cells do that is by using messenger RNA, mRNA. And so, as with, very similarly to the process of DNA replication, the double-stranded DNA will have to be separated. In this case, you see that we have a little bubble that is formed. It's called a transcription bubble, whereas transcription is actually the production of RNA from DNA. Then what will happen is your RNA polymerase, which is the enzyme right here, just like a DNA polymerase, will tie nucleotides together, but you get the same kind of principle. The template is the DNA, so that's going to be one set of bases. And then RNA nucleotides are going to be put on according to the complementary sequence. So you can see there's an A for that T, the C for G, G for C, U for A, C for G, etc. And so the polymerase is just going to move along the gene, and it's going to keep adding. So it'll add an A here because of that T, then it'll add a C because of that G, it'll add a G because of that C, and it'll keep going until it gets to the end. And that's something that's pretty important to think about with genes, is that genes have beginnings and they have ends. So they have a place where the enzymes know where to start making the RNA, they have a spot where they know to stop making the RNA. And the idea is that RNA will start at point A, end at the end, at what we call the terminator, and that will be that one piece of RNA that will then be processed a little bit, and will move off to the ribosomes to be translated. Now, I do want to very quickly go over and talk about one thing that you tend to see with eukaryotic RNA that you don't really see much with prokaryotic, but that is we've got little bits of our DNA, a message inside the DNA that doesn't actually, it's like junk DNA. It doesn't get expressed in the final protein. It'd be kind of like if you were reading instructions for how to build something like a desk, and for some reason they had stuck in some instructions for how to build a TV stand. They don't really apply to the task. And so we call these things introns. And so the introns, as you can see, there's one here and there's one there, are the junk DNA that is not going to be used. And again, this is, so far as we know, is only in eukaryotes. But the cells have a way of cutting that out, and that's what it's kind of showing here. So even though this is what the DNA looks like, an mRNA strand will be made directly off of the DNA, so that will include the sequences that have the introns in them. But as you can see, those introns are being cut out by enzymes, and what we're left with is an mRNA that has just the other parts, the exons, which are the valid instructions. Then this is the mRNA that will leave the nucleus and go to the ribosomes to be expressed.